series in food and health. And we've put these on to provide you with really good up-to-date scientific evidence on topics that we know are of real interest to you. And today, I love the topic, balancing bugs, the role of good and bad microbes in our food. We've a lot of interest in this one, and I think it'll be really interesting to learn so much about it. So we've two speakers this evening. We've Dr. Garino Macari and Professor Shay Fanning. So Garino is an assistant professor in molecular microbiology and applied bioinformatics in the School of Biology and Environmental Science at UCD. And his research focuses on microbial genomics, antimicrobial resistance, and I love this, microbial dark matter using multi-omics integration. Okay, so he leads many international and national projects, supervises PhD students across themes of One Health, and he contributes to the WHO, EFSA, and Microbiology Society initiatives. And he's joined today by Shay, who I'm delighted to welcome. And Shay is Professor of Food Safety and Zoonosis at UCD. He's director of the UCD Centre for Food Safety, and he also helps me a lot as Deputy Director of the Institute of Food and Health. So Shay's research focuses on bacterial genomics and understands bacterial risks to One Health associated with antimicrobial resistance, and at the genetic level of the characterization of zoonotic foodborne pathogens of importance to human health. So that's a big mouthful, but Shay will translate all of this for you. And Shay, like Garino, is a member of several high-level scientific committees, including the Scientific Committee of the Food Safety Authority of Ireland. He works with the WHO, the FAO, and many more. And he's an elected fellow of the American Academy of Microbiology. So I'm sure these two are going to give us a huge amount of information. We're going to learn a lot, and I hope have a bit of fun on the way. So during the talks, if you have any questions, please add to them questions and answers tab that you'll see at the bottom of your screen. At the end of the talks, we'll go through those questions and I'll pose those ones to Garino and Shay, and we'll have that answer session at the end of the lecture. Okay, so I'm delighted once again to welcome you all, and I'll hand over to you both now. So Garino, I'll let you start. Thank you very much.
Okay, thank you so much for the invite, first of all, and for the kind introduction as well. As mentioned, my name is Dr. Garino Macari. I'm delighted to join you tonight. I will talk about the role of good microbes in my part, so the tiny organisms that live in us and around us as well. Share a little bit of examples from my own research into how they shape our food and our health. Now, I work at the School of Biology and Environmental Science at University College Dublin. My background is in food safety and pathogen genomics, but over time I become fascinated not only with the microbes that are pathogen for us or they harm us, but also those that help us somehow. So tonight, we'll focus on these allies, the microbes we actually want in our lives and in our food as well. And then we will see as well with Professor Fanning how and they can be as well dangerous. So we go before we go further, and I want to have time at the end to thank. I want to really acknowledge the colleagues who contributed to the studies I'll present today. Professor Evelyn Doyle and Professor Seamus Fanning at UCD that will present later again, Professor Paul Cotter at Chagas and APC Microbiome Ireland, Dr. Aaron Walsh and John Leach, who contributed on the case studies I will introduce later. Their expertise, as usual in science and teamwork as well, made all the research I'm talking about possible. Now, my research uses DNA sequencing as technology to study what we call microbial communities. We use high throughput sequencing and integrated multi-omics approaches, and we can uncover and use it for discovering how these microorganisms evolve, adapt, and interact as well to each other. This applies not only to pathogens, but as mentioned, also beneficial microbes that we previously knew very little about. Much of my work involves developing new tools to detect, in particular, track these microbes, including the unknown ones that could emerge in the future, and we sometimes call them disease X. By combining all this information, all these technologies, and in particular metagenomics, and I will talk about that in a few slides, we can design as well diagnostic tools to prepare for outbreaks in humans, animals, and even plants. Now, think of it as a building early warning systems that's a tool somehow that can detect and threat before it spreads. But beyond pathogens lies what we call microbial dark matter, as was mentioned earlier, which is the vast diversity of microbes that we know that are present or that they can be in some systems, but remain uncultured or they're not discovered or not culturable yet. We can use innovative models like fermented foods, and we can explore in this way this world. And fermented foods, as I will mention later, are particularly useful because they are rich in microbial diversity, but also very easy to sample and have in the laboratories and use them as models as well. However, remember, microbes are invisible, they are incalculable, but potentially very useful for many of the strategies that I listed there in the slide. Some of these microbes are also called most wanted taxa, which means that we know they are in the environment, but we haven't been able to culture them yet. These microbes could be used in the future for health, agriculture, or biotechnology as well. Now, microbes can be both beneficial and harmful, as we will see as well later. On one hand, they improve digestion, enhance nutrient absorption, create flavor and texture in food. On the other hand, they can contaminate food, producing toxins or triggering maybe allergies. And my interest lies right there in the middle, maximizing the benefits of these microorganisms, or microbial communities, while minimize the risk of infection and for topics interesting for safety as well. Now, what if I told you that your body carries more bacterial cells than human ones? Now, this simple fact highlights how we are connected with microbes in general. So in fact, even a newborn baby carries around 10 to the 13 microbes that are more than the number of stars in our galaxy. So from the very beginning of life, we are microbial microorganisms. So tonight, I show you how these microbes are not only good, but also essential, especially when it comes to food. Now, we define good bacteria, and these are a little bit of introduction and definitions for tonight. So commensals are the one that live in or on us without causing harm. We have as well mutualists, which are the bacteria that, or microorganisms in general, that benefit us while we provide them with a home. After they need to be in the system with other microorganisms, but the microbiome is more than just a list of microbes, as we can see as well when we analyze these foods. It's an interlinked ecosystem of genes, networks, and interactions as well. Now, even our own bodies carry multiple microbiomes. So it's not correct when we say our microbiomes is made of these microbes, because we can have microbiome specific in the gut, of course, in skin, but also mouth and many other places in our body. And food, of course, carry their own microbiomes. So wherever we are looking to a baby, a piece of cheese, or a jar of sauerkraut, microbes are everywhere, and there are different ways to describe them and different technologies as well that I will try to mention later on. Now, good microbes play three main roles. So the first, very important, is digestion and metabolism. So they break down fiber into what we call short-chain fatty acids like a butyrate, which feed our intestinal cells and even protect against inflammation and potentially as well as against cancer. The second very important piece is the immune system training. So in fact, from early life, microbes train our immune system to distinguish friend from foe and shaping as well lifelong immunity. The last piece I wanted to introduce is how they protect against pathogens. So they produce, in fact, some of them antimicrobial substances and crowd out harmful microbes. For example, lactobacillus can prevent candida from overgrowing. Or in the example here in the figure, I presented how the heterolactic fermentation from lactose to glucose and galactose produces as well lactic acid, CO2, ethanol, but also antimicrobial molecules like, for example, lactobacillus paracasi or plantarum that produce specific molecules that are against other microbes, or bifidobacterial longum that produces as well lactic acid. And beyond that, microbes contribute to other functions. We know now that are involved in what we call the gut-brain axis for mental health, but also vitamin production, for example, vitamin K or B12, and of course, skin health as well. Now, fermented foods are one of the best examples of how humans have long harnessed good microbes. So fermentation is, in fact, very ancient, but modern in the terms of products that now we can find as well in our in the shoppings, in our fridges as well. In general, the action of these microorganisms is to preserve food, and this modality of preservation is also to use for enhancing the flavors and produce metabolites that may support health. So the microbes in fermented foods depends on the substrate. Think about milk, vegetables, or sugar-based food, and the method how they are produced. Some of the products are produced with starter culture, or we can also have a spontaneous fermentation, and also the environment. So we are we know now that the local conditions and geography as well gives a specific feature on food and fermented foods in particular. What we know though is that brine and sugar fermentation like sauerkraut, kimchi, and kefir contain more diverse microbes than dairy fermentations. Now, let me show you one of those stories from my own research in Ireland. As we will see in the first case study, for this particular study, I'm bringing you with me to a quick Irish trip started in Cork, driving and meeting local small producers, small farms, along the west and south coast in the middle of the country, including small villages of farmers who speak only Ascuelga. But when standing in front of food and cheese, there were no languages barriers at all. And as you can see in the pictures, including the hygienic ones that might be translated then in Food Van Outbreaks, and Professor Fanning later on can tell us all about. In total, for these studies, I collected 55 different artisanal cheese samples, visiting 27 different farms around the country. And we created as well a database including other 104 cheese samples from other datasets, always related to Ireland. Now we sequenced the DNA in cheese samples, capturing microbes from bacteria, yeasts, and viruses as well. And in total, we detected 23 main microbial species across many samples. So we are talking about 184 cheese samples. That might sound like a very small number, especially compared to the thousands we see in the human gut. But here, it's not about how many microorganisms or that we can find or describe. It's about which ones are present and what they do there. Things like a little bit like a recipe, and you don't need several ingredients or hundreds of ingredients. What matters is the right one that gives the specific flavor, or the right amounts that gives the final product as well. We then started, for this particular story I wanted to tell you tonight, the microbial abundance correlated with the production of volatile compounds. These are the molecules that give cheese its aroma and flavor as well. We found that just 4 microbial strains were significantly linked to the differences in terms of flavor profiles. So not all microbes matter equally in the same way, but sometimes even a few strains can define the whole taste experience of that specific food or product. So now next time you enjoy a piece of cheese, remember you're really tasting the work of these microbes, even in the small amounts of their presence and their content. The strain level, in fact, differences become even clearer. So you can see in the pictures in the left-hand side, we have 2 brevibacterial linens, in particular strain 1 and strain 2, that strain 1 was positively linked to certain favorite flavors, while another strain, on the other hand, was actually negatively correlated to the presence of flavor in that food, in that cheese. The same pattern we found it for Streptococcus thermophilus, but also Lactococcus lactis. So even within one species or one single species of microorganisms, the strain can behave very differently, some enhancing the flavors, or other reduce the flavors, or give even a better effect on the community, on the texture, on the final aspect of the food. Now, we always say with the students, it's like having two different cousins in the same family, where one is an amazing cook, but the other burns the dinner. But in reality, they are very close and related genetically as well. Now, microbes also contribute to the appearance of cheese. So we identify genes, in fact, for carotenoid production, that natural pigments that give the cheese their yellow, orange, or even reddish tones. And across the 58 genomes that we built starting from the original DNA of the cheese microbiomes, we found more than 246 potential genes involved in the biosynthesis of these carotenoid. So the color of your cheese isn't just from the tradition, it's also literally painted by microorganisms. We found in particular these two microbes, so Brevibacterium and Glutamicebacter, that carry these pigment genes. And they were capable of producing at least 14 different carotenoid compounds, including lycopene and neurosporin as well. And these pigments don't just change the color, they might also have properties like antioxidant properties or adding subtle health benefits. Now, these microbes also carry genes for bacteriocins, and these are natural antimicrobial peptides. These act like microbial weapons from some of the microorganisms that prevent unwanted bacteria from growing. So in every block of cheese, microbes are quietly policing their neighborhood, keeping pathogens in check. Now, just to sum up a little bit about this study, from this one, we learned that cheese microbes contribute in three major ways. We have flavor, color, and protection. So even a relatively small set of microbes can create a remarkable diversity in terms of taste, safety, but also quality of the product. And this is just one food system. Let's now move to another wider view of fermented foods, including vegetables and drinks as well. In fact, in this study that I want to present tonight for the last few slides, I wanted to take you beyond cheese and into the world of fermented vegetables and drinks, for example. And in this study, we look in products like sauerkraut, kimchi, kefir, kombucha, miso, and tempeh. So you might have some of these in your fridge, or maybe you've heard about it. And if you do, you also, especially if you have some of them in your fridge, you're also storing a living community of microbes just in your fridge. In this specific study, we analyzed 58 fermented foods in total. So the brine, sugar-based fermentation such as sauerkraut, kimchi, and kefir show the highest microbial diversity, in particular dominated by lactobacillus, leuconostoc, and bifidobacterium that maybe you're familiar with as well. This diversity is not random, and it reflects how the substrate, so the nature of that specific food, for example, cabbage, milk, or sugar water, in the case of water kefir, shapes the microbial community. And when we compared the fermentation methods, we were able to see clearly that the spontaneous fermentation, the one that rely on the natural microbes present in the raw material, were even more diverse than those using starter culture. We were expecting that, but we demonstrated as well in terms of scientific evidence. Now, these rich microbial communities also carried a lot of genes linked to health benefits. So then, if you think about the variation of these microbial communities, it's very important as well to remember how the potential benefits they might bring. So in other words, the diversity isn't just the flavor of these products, but also functional. So we know as well that what they bring in terms of information in the genomes and in the products. Now, just to conclude this presentation for tonight, I wanted to highlight how modern technologies, in particular DNA sequencing, we can use it for map these beneficial microbes down to the strain level. So we are able to detect not just the species present, but also their specific genomic address. And this means as well that we can, in future, design or select microbial communities, improving flavor, safety, health outcomes, as I highlighted earlier. So fermented foods in general then are not just old traditions or something that we can find or produce in our bench in the kitchens, but they are living ecosystems. They are created by culture, but now we can refine, and they are refined by science. And of course, it's not always easy to classify a microbes when it's good or bad, or even some usage of antibiotics can disrupt beneficial communities, as you are aware. And probiotics in general are not magic pills, so there's not just one context where you can take a pill and magically change the function in your gut as well. There is also this conception that taking probiotics will definitely save some from bad feeling, but sometimes, of course, it helps. Sometimes we need to be clear on the fact that uh, there's no one size that fits at all. In particular, you can see that just going in the strain level, we can find microorganisms that are or not active against some specific effect. Now, without good bacteria, though, we simply wouldn't function. So we know that, and we couldn't digest food, fight infections, or even develop properly. In many ways, as I mentioned at the very beginning, when I compare the stars in the galaxy and the babies, so you can see that we are superorganisms, not in spite of the bacteria, but because of them. Now, good microbes are everywhere. They're found in our plates, guts, and at the art of food innovation. And with that, I'll hand over to my colleague, Professor Shannon, who will now introduce the darkest side of microbes. So what we call them the bad ones. And if tonight I've shown you why some microbes are essential allies, the next talk will show you maybe why others keep us awake at night.
Thank you so much, Garina. Some really lovely pieces of information there and really well explained. I'm going to hand over to Shay now, but just to remind everybody, if you have any questions, pop them into the Q&A and we'll come to them at the end. So thank you very much. And Shay, I'll hand over to you.
Thanks, Eileen. And I hope everyone can see my slides. And again, I'd like to welcome everybody listening to us in this session for this evening. And I hope, as Garino has said, that you find it interesting and full of new things that perhaps you didn't know before. So you've heard about the very good bugs. Now, unfortunately, we turn to the bad bugs, and I want to focus on bugs that are associated again with food that cause foodborne infectious diseases, and tell you a bit about how they might challenge food safety. So if we take a look back over the past 40 years or so, and we just look in terms of the landmark events in infectious disease emergence, there's been a couple of notable things, some of which we will undoubtedly remember, particularly in the last number of years with the outbreak that we all remember very fondly as the COVID-19 pandemic. But if you go back further in time, there's a number of other microbiological hazards, shall we call them, that also have a major impact on public health. Particularly, for example, if you look at 1982 and the emergence of a particular type of E. coli, and we generally tend to think of E. coli as a good bug, so it would fit within Garino's portfolio, but this one, as they say, is a good bug gone bad. And then there's other examples, the other two stars in this particular slide, which illustrate two types of influenza which have become associated with the food chain. So I suppose to summarize these landmark events, we would say that diseases which are transmissible between our animals and us as humans, we call these zoonotic diseases, and they're certainly a significant challenge to human health. But did you know that more than 60% of these infectious pathogens have their origins in animals? And more than 75% of all emerging infectious diseases are zoonotic in origin. Therefore, zoonotic pathogens are really important to food safety. So it begs the question as to what is food safety and how do we describe it? I suppose it's not exactly a subject that you find on the curriculum anywhere described in any great detail. It's kind of an amalgamation of different things. But if we were to try to define food safety, then what we would say might be the following, that we would consider it as an application of scientific-based methods, so much like what Garino has described in his very nice talk, But these methods are focused on the characterization of different kinds of hazards that can cause us harm. And these hazards are generally biological, chemical, and physical. And here in UCD, we mostly focus on the biological hazards in my lab. And so we try to develop an understanding of them so that we can then begin to control them through regulation. And the purpose, of course, of that control is to protect public health. So it's really applying scientific methods, much like what Garino was talking about, to try to understand these hazards and then to control them. There are different kinds of foodborne illnesses, as we may know. There's what we would call infection. And this is where we have pathogens that are alive in the contaminated food which we may ingest. And then these bacteria develop in our gastrointestinal tract, and then they may or may not cause signs of illness. And usually infections take a couple of days to manifest. On the other hand, you may have an intoxication, and this is where you have a poisonous substance which is present in the food again at the time of consumption. And this poisonous substance is probably the result of a toxin that's produced by a bacterium. A typical example, of course, being foodborne botulism. And again, this has a slightly different presentation in the sense that it takes only a few hours to develop clinical signs. And there are a number of others as well that we think about from time to time. In terms of how these infectious agents, as I mentioned at the beginning, get transmitted, well, we need animals for that. And there are some examples, and I just picked out a couple here just to illustrate the case in point. Bats, for example, are increasingly being recognized as a source of different types of coronavirus. In fact, the COVID-19 epidemic was caused by a virus called SARS-CoV-2, which can be found in bats. And similarly, if you look in the middle of my slide here, you have a very unusual example of a roundworm that was found in a lady in Australia about 2 years ago. And this is a very interesting link to food safety because in this particular case, the roundworm, which is normally associated with carpet pythons, which live in grass, became lodged in the lady's brain. And you can see an image of her brain with the occluded area, which represents where the roundworm was found and removed from her brain. And so the link to food safety here was that the lady was very interested in using grasses, which she found locally near her home. And she used these grasses in different food preparations. And of course, the grass was contaminated with the roundworm, which was secreted originally by the carpet python. And then on the right-hand side of my slide, you have the more typical vectors that I'm going to talk about, which are colonized food-producing animals, which are in fact recognized as reservoirs of some of these very bad bacteria. Here are a couple of examples for you to look at. So on the top part of my slide here, you have a cow, and you can see this little child drinking milk directly from the udder of this cow. And of course, we know that cattle are reservoirs of pathogenic types of E. coli, the one that I mentioned earlier on in the earlier slide, also Salmonella typhimurium. And in our avian population, you see here these birds that are housed in flock rearing houses. These are colonized by other types of pathogens or bad bacteria called Campylobacter and other types of Salmonella, in this case, Salmonella enteritidis. But it's not just the animals that we have to be concerned about. We also find some of these pathogens on foods that we directly consume without cooking. So for example, in the picture that I'm showing you here of the tomatoes, they look perfectly nutritious, but they potentially could be contaminated with certain types of bad bacteria, if you like, because of the way these fruits or produce were grown in the field. And then in 2011, we had a spectacular outbreak which was centered around Germany, and maybe some of you remember this, where there was a very unusual type of E. coli called E. coli 0104 that emerged. And this was associated with contaminated bean sprouts. And again, these bean sprouts look utterly nutritious. We don't really cook them. We tend to eat them raw. And in this particular example, this bacterium contaminated these bean sprouts, which caused several thousand people to become very ill very quickly. And about 55 of these people died, mainly in Germany, as it turned out. Now, Garino mentioned antimicrobial compounds and the fact that some of these good bacteria produce these very same compounds. And in fact, we've known this for a very long time. However, our use of antimicrobial compounds to treat bacterial infections has resulted in a situation where many of the bacteria pathogen list. So that's what you're seeing here in this particular slide. This list really cataloged the sort of first 26 of the most important bacteria that needed to be controlled by way of trying to preserve the efficacy of antimicrobial compounds. And because there's a race between us and the bacteria in terms of trying to develop new antibiotics, in the past 7 or so years, 13 new compounds have been put into clinical practice. And this has caused a revision of this list very recently by the WHO, and you can see this on the right-hand side. And if you look at the stars on the left and the 2 stars on the right-hand side of my figure, these indicate bacteria on this list that are relevant to food safety. So if you look at the red star down towards the bottom on the left side, you'll see non-typhoidal salmonella that's resistant to a compound called fluoroquinolone, a very important antimicrobial compound when it's for use in the treatment of salmonellosis in certain cases. And then if you look at the gray star, which is the top left-hand side, you see another example of an Enterococcus faecium, which has become resistant to vancomycin, a type of antibiotic used in clinics. And these 2 pathogens, if you look on the right-hand side of my slide in 2024, have now moved position, and they've become more prominent. In other words, of greater concern to the WHO in terms of our ability to continue to treat these types of infections. Other pathogens are listed in the 2024 list, and I'm not really going to deal with them for the purposes of this evening's talk. One of the other aspects which is also of concern to us, not alone with respect to the use of antimicrobial compounds, but it's the effect that we're trying to understand by way of climate. And a systematic review of literature done very recently estimated that half of all infectious hazards are aggravated in some way or other by climate change. And we really don't have a great understanding of this at the moment. If I try to break this down for you and try to simplify it, at least as best as I can understand it, we know that climate change is giving rise to increases in greenhouse gas emissions in the atmosphere. And we know that that's causing a warming of our atmosphere. So much so that if we try to translate that climatic effect in terms of what it might be doing to bacteria, if we look on the bottom of this slide, this is a typical bacterial growth curve over time. So it simply measures the increase in bacterial numbers with time and available nutrients, very much like the fermentation processes that Garino was explaining in his talk. Now, if you look at the orange line, and let's take that as a typical example where bacteria are growing at a temperature of 37 degrees. And let's assume a climatic influence here by way of an increased temperature. So that increased temperature will pull that growth curve to the left-hand side. So if you look at the left-hand side, you'll see a green line with dots. And this would suggest to us that the heating of our atmosphere will cause our bacteria to have a much shorter lag time and a much faster growth time as the climate begins to heat up. So it does cause an impact on what bacteria can do, and it will also cause an impact on the microbiomes that Garino was very nicely explaining. Now, this is happening all over the world, and in some parts of the world, bacterial foodborne illness is a much more serious public health challenge than in other parts of the world. And in this world map here, you can see a summary picture of that, so much so that the green parts of the world are where foodborne illness is of greater consequence compared with other parts of the world in blue, where it's of lesser consequence. But nevertheless, irrespective of where you are on the planet, the same types of pathogens are causing concern to public health, and these are regarded as enduring bacterial foodborne hazards that need to be regulated by way of developing an understanding of them and then translating that understanding into a regulation that helps us to protect public health. So what does the Irish picture look like? Well, our colleagues in the Health Protection Surveillance Centre on Sheriff Street in Dublin collect information each year that describes the number of cases of gastroenteric or zoonotic diseases that are reported through the clinical reporting systems in our country. And here we can see two categories. One category which we would describe as infectious diseases, like the ones I described in my definitions very early on, and others which we describe as intoxications, where it's the toxin produced by those microbes that are of concern to us. And if you just look at the top part of my slide, which describes the bacterial infectious intestinal diseases, you can see that the number one bacterial hazard is Campylobacter infection. And this is caused, as I've said before, where this bacterium is transmitted from reservoirs in food-producing animals. These are our chickens, our ducks, our geese, through the food chain to susceptible humans where they cause infection. Now, if you look at quarter one and quarter two in the year 2024 and the year 2025, the current year, you'll see the numbers of cases of Campylobacter infections that are reported. And again, if you do a similar exercise right throughout this table, you'll see that those cases are pretty much the same. So our epidemiology appears to be relatively stable over time, except perhaps in the case of the listeriosis number. And this is what we saw in quarter one 2024. We had 5 cases. Quarter one 2025, we had 2 cases, and I'll come back to that later on towards the end of my talk. As I mentioned, foodborne illness is caused by the consumption of contaminated food. And in fact, if you think about our alimentary canal, which is what this picture on the left-hand side is showing you, there's lots of challenges that are faced by foodborne bacteria when they enter the human domain. So as our bacteria, if you like, transit from the contaminated food down into our gastrointestinal tract, they have to overcome challenges such as low pH environments that you find in the stomach. They have to overcome challenges such as the antibacterial effects of bile salts, the lack of oxygen in the gastrointestinal tract, and of course the battery of immune cells that are designed to attack them. And again, as Garino mentioned in his talk, bacteria tend to fine-tune this immune strategy for us. We also are thankful, I guess, or we have to be thankful for a good immune system because it does protect us. However, individuals have different vulnerabilities to zoonotic hazards. So for example, in some of us, a bacterial infection like I'm describing might cause a mild illness, as you can see illustrated rather graphically on the left-hand side. Or it might be more serious. If the illness is more serious and it affects a more vulnerable member of our family or of our community, we could end up dying as a result of an infection. So to protect public health then, we must be able to track these infectious foodborne bacteria. And this alludes to something that Garino mentioned during his talk. In order to do this, we must be able to determine their DNA sequence or their genetic blueprint. And we use that genetic blueprint to then track these bacteria of importance to public health. And the way we do this is we simply isolate DNA from a pure bacterial culture, and we then sequence this DNA such that it gives us a complete bacterial genome. So that's a complete picture of what that bacteria may be able to do and how it may be able to cause harm to a susceptible individual. Now, I mentioned tracking these pathogens right around the world, and there's a database called the Genome Tracker Database, which was developed in 2013 by our colleagues in the FDA in the US. And since this database was developed in 2013, as you can see from this graphic, there's been an exponential increase in the number of bacterial genomes that are deposited into the database. Now, this does a number of things for us. It allows us to analyze these databases very quickly and in order to compare isolates that are of interest to us versus isolates that are already submitted to this database. And the other key thing that this database helps us to do is it helps us to identify outbreaks even before we begin to see signs of those outbreaks happening in the broader community. The database works by being able to submit bacterial whole genome sequences into these 3 database servers, the NCBI database in the US, the EMBL database in Heidelberg in Germany, here in Europe, and the DDBJ is a database in Japan. And these databases then synchronize information every 24 hours such that anybody anywhere in the world can access these databases, go in and interrogate them, and try to identify things that they may be interested in. So much so that laboratories around the world contribute genome sequences to these databases in order to help the public health effort to tracking and controlling bacterial infectious agents as they track around the world in our food supply. And if you look at the bottom right-hand side of my slide, you can see a picture of the British Isles and a dot which lights up Dublin, and that's associated with our laboratory here at UCD in the Centre for Food Safety. So for the last couple of slides, I just want to turn my attention to the application of what I've been showing you. And I just want to take a case study focusing on Listeria monocytogenes with deliberate intentions, as you'll see. So what I want to look at here is to ask the question, what does a Listeria outbreak look like? And I also want to make a comment on the ongoing outbreak that's associated with ready-to-eat meals here in Ireland. Now, Listeria monocytogenes is a gram-negative bacterium. It's one of the bad guys. And it's a bacterium that tends to have a ubiquitous distribution. So we find it in lots of different settings. For example, we can find it in our food-producing animals. We can find it in the soil, water, and farm environments. We find it specifically in food processing equipment and in our food production environments. And we can also find it associated with certain types of foods, either of animal or non-animal origin. But the interesting thing about this bacterium is that whilst generally in the environment we would describe it as a saprophytic bacterium, when it gets close to us as humans, it can become a really serious facultatively intracellular bacterial pathogen. And again, surveillance studies tell us that in the EU in 2023, there were almost 3,000 cases of listeriosis reported through the various member state surveillance systems. And of those cases, almost 20% of those individuals died. So this is a very serious example of a foodborne infection. Not alone can Listeria monocytogenes adapt to the human condition, if you like, but it also can be found, as I've mentioned, in the food and the production environment. And if we think about some of its attributes, then we may be able to understand why it survives in food and in the production environments. For example, Listeria monocytogenes is able to grow at refrigerated temperatures. Now that may seem counterintuitive because we use fridges in our homes to prevent spoilage of our food and also to prevent the growth of some of these pathogenic bacteria if they are present in our food. But Listeria ignores this type of logic and is able to grow at 4 degrees. So we call this bacterium a psychrophile for that reason. It can also form biofilms. It can become resistant to biocides. So biocides are those solutions or sanitizers that we use to clean surfaces and that the food industry would use in its production settings in order to keep hygienic standards as high as possible. And again, rather like Guerino has said, when you think about fermentation and what fermentation does, many of these fermentative processes impose a stress on these types of pathogens, which negates their growth. But in the case of Listeria monocytogenes, it's able to respond genetically by offering countermeasures to things like low pH and high salt concentrations. So that epidemiologically, when you look at where we find Listeria monocytogenes, it's found in a wide variety of foods, food processing environments, and typically the foods that are associated with contamination outbreaks linked to this bacterium are things like ready-to-eat delicatessen meats, raw meats, soft cheese, and seafood. So the real question here is, how does Listeria adapt to these different types of environments? And the answer to that is that we only know part of that answer at the present time. So much so that when these controls that I talked about fail, you then get outbreaks. And this slide that you're looking at here is simply a synopsis of one of the largest listeriosis outbreaks that was recorded, and it was recorded in South Africa between January 2017 and July 2018. And in this particular outbreak, which was attributed to this type of delicatessen meat that you see here on the bottom left-hand side of my slide, there were over 1,000 laboratory confirmed cases of illness, with 216 deaths attributed to the consumption of this contaminated delicatessen meat. And when you look at this epidemiologically, there was just one type of Listeria monocytogenes that was involved in this outbreak. In other words, a single unique type of strain that caused the outbreak. And again, like Garino said towards the end of his talk, that's where whole genome sequencing becomes really important, helping us to detect these international outbreaks and to try to control them. Another example of an outbreak which happened in Manchester in 2019 that I just want to highlight was associated with an old people's home where there were 9 human cases of listeriosis reported over a 2-month period, and 6 of those 9 cases unfortunately succumbed. And the reason those older members of society became ill and died was because they consumed contaminated sandwiches. These sandwiches were contaminated with Listeria monocytogenes. And again, the outbreak investigation here shows you very nicely, if I can just use my cursor to highlight it, if you can see it, this is a very tight outbreak up here where all of these isolates that came from clinical cases of infection, that came from the contaminated food and the processing environment were clearly linked. And that outbreak is quite distinct from other similar types of Listeria monocytogenes, which are not epidemiologically linked to that particular event. And that brings us then closer to home. And as you know from the media, going back to August of this year, there were reports of listeriosis cases associated with a company that was producing ready-to-heat meals. So I'm not involved in this particular outbreak, but from what I know, this company has been in production for quite a long time, very good reputation, and unfortunately something went wrong, which gave rise to a recall of the products that they were producing, and a number of listeriosis cases being reported through that surveillance system that I mentioned earlier on, and regrettably one of these individuals died as a consequence. So what are the at-risk groups that we find in these cases of listeriosis infections? Well, there's essentially 2 different presentations of this infection. One is a non-invasive form, and the second is a very invasive form. And the invasive form is the one that I'm highlighting here, whereby you have listeriosis associated with particular human at-risk groups. And those would be individuals in society who might be immunocompromised or who might be undergoing cancer treatment, the elderly, of course, and pregnant women. And in this case, the manifestations of listeriosis is either as a form of adult listeriosis, where the central nervous system may become infected and you may get a meningitis or a meningoencephalitis, or perinatal listeriosis, where this particular bacterium has the ability to cross the placental membrane, and it might give rise to an abortion or a stillbirth or an infected neonate. As I mentioned several slides back, this is one of the more serious presentations of a foodborne illness, and the reason this is particularly serious is because you have very high mortality rates, particularly in the invasive cases within these at-risk groups. Most of this happens as a consequence of the consumption of contaminated food. Now, when you look at the epidemiology of these listeriosis infections, in fact, you see something very interesting as well. So if you look at the red bars here on my slide, these are what we call hypervirulent clones of Listeria monocytogenes. There are certain strain types that we can recognize by whole genome sequencing. And if you look, there's a preponderance of these particular strains associated with dairy products. And that's why it's particularly important to advise women, if they're becoming pregnant or if they are pregnant, to avoid the consumption of unpasteurized dairy products, unpasteurized cheese, unpasteurized milk, and similarly seafood. In contrast, if you look at the blue bars here, these are lesser virulent, or what we call hypovirulent strains, different kinds of Listeria monocytogenes that are associated with food or the food processing environment. And in these cases, these are less likely to cause infections in humans, but they're more adapted, if I can put it like that, to the food processing environment. So using whole genome sequencing helps us to recognize differences between these types of Listeria monocytogenes, and therefore we can begin to think about stratifying the risk to human health. What you're seeing here is undoubtedly evidence of what we call clonal adaptation to specific environmental niches, and this to some extent helps to explain some of these observations. So to try to sum up then, I'm painting you a very bleak picture with respect to the bad bugs, whereas Garino is giving you a very optimistic view of this. Food by itself has its own microbiome, as Garino mentioned, and the predominating bacteria within that food microbiome are good bugs. Occasionally, bad bugs get to contaminate the food, and therefore, to try to protect ourselves, there's a couple of key things that the World Health Organization tells us to do. When we're preparing food, we keep our hands and utensils clean. We separate raw food from cooked food. We cook everything thoroughly. We keep food at safe temperatures, like the refrigeration temperature that I mentioned, being wise to the fact that Listeria monocytogenes may cause a problem. And we use safe water and safe raw materials. And with that, I'll thank you for your attention.